This video finishes up Chapter 12 on biosignaling. In the last video, I mentioned that there are four major types of receptors: G protein-coupled receptors, also known as GPCRs, receptor enzymes or enzyme-linked receptors, gated ion channels, and nuclear receptors. In this final video, we will talk about some enzyme-linked membrane receptors as well as gated ion channels. Many membrane receptors consist of an extracellular ligand binding domain as well as an intracellular, so within the cell, catalytic domain. And the most common type have tyrosine kinase activity. Let's break down that word, tyrosine kinase. So a kinase is a type of enzyme that catalyzes the transfer of a phosphate group from ATP to a specified molecule. So to have tyrosine kinase activity means that we're going to transfer a phosphate group to a tyrosine residue. It will add the phosphate group to itself, also known as autophosphorylation, and this leads to a conformational change allowing binding and catalytic phosphorylation of specific target proteins. And it will add a phosphate group to a tyrosine. In addition to this, there are some catalytic domains that have guanylyl cyclase activity. So let's break down that name. So cyclase is a name for a type of enzyme. Remember, enzymes often have names that end with ASE, and it's going to catalyze a chemical reaction to form a cyclic compound. And with guanylyl cyclase, it's going to convert GTP, guanosine triphosphate, to cyclic guanosine monophosphate. There are a variety of receptor tyrosine kinases, also called RTKs for short. What these receptors have in common is that they initiate signals through tyrosine kinase activity. And these include insulin receptors, INSR, the vascular epidermal growth factor receptor, the platelet-derived growth factor receptor, the epidermal growth factor receptor, the high-affinity nerve growth factor receptor, and the fibroblast growth factor receptor. What these have in common is they all have a tyrosine kinase intracellular domain, which is on the cytoplasmic side of the plasma membrane. And then the extracellular domain varies from receptor to receptor, reflecting the different growth factor specificities. And these extracellular domains, shown on this side, are typically combinations of structural motifs such as cysteine or leucine-rich segments and segments containing one of several motifs common to immunoglobulins. Here are two examples of receptor guanylyl cyclases. In the receptor shown on the left, this is a type of homodimer with a single membrane spanning segment shown here. And this connects the extracellular ligand binding domain to the intracellular guanylyl cyclase domain. The type of ligand that this might receive is ANF, which stands for atrial natriuretic factor, ANF, which is released by cells in the cardiac atrium when the heart is stretched by increased blood volume. Carried in the blood to the kidney, ANF activates guanylyl cyclase in cells of the collecting ducts. A similar receptor guanylyl cyclase in the plasma membrane of epithelial cells lining the intestine is activated by the peptide guanylin, which regulates chloride secretion in the intestine. And this is the receptor which is also the target of a heat-stable peptide endotoxin produced by E. coli and other gram-negative bacteria. The other type of guanylyl cyclase is a soluble heme-containing enzyme that is activated by intracellular nitric oxide. And this form is present in many tissues, including smooth muscle of the heart and blood vessels. We're going to illustrate a receptor tyrosine kinase by talking about the insulin receptor. Insulin is a peptide hormone that is produced by the beta cells of islets of Langerhans and the pancreas. Insulin reaches target cells such as the liver, muscle, or fat tissue via the bloodstream. And the binding of insulin to the insulin receptor initiates a cascade of events that ultimately leads to increased glucose uptake. And diabetes is a disease where you're unable to make or sense insulin. Now, normally, glucose transporters are stored within the cell membrane in vesicles. However, when insulin binds with the insulin receptor, these vesicles move to the surface and fuse with the cell membrane, increasing the number of glucose transporters in the plasma membrane. And then when the insulin level drops, these transporters are removed by the plasma membrane by endocytosis, forming small vesicles. And then these smaller vesicles fuse with a larger endozyme, and patches of these endozyme enriched with glucose transporters bed off to become small vesicles ready to return to the surface again when insulin levels rise. So let's focus in on the insulin signaling cascade. So here we have the receptor. So this is a receptor tyrosine kinase. And the first thing that's going to happen is the ligand of insulin will bind to the extracellular domain and activate the catalytic domain on the inside of the cell. The catalytic domain in one receptor phosphorylates tyrosine residues in another receptor. And this receptor autophosphorylation allows binding and phosphorylation of protein IRS1. IRS1 stands for insulin receptor substrate. And this picture here just shows the inactive unphosphorylated TK domain, where this shows what the active triple phosphorylated TK domain looks like. So here we see the entire cascade. So the insulin binds to the receptor, and then these two catalytic domains get triply phosphorylated. And then as a result, these will phosphorylate an insulin receptor substrate 1 on its tyrosine residues. Then the phosphorylated IRS1 binds to the SH2 domain of the GRB2 protein. SH2 is an abbreviation of SRC homology 2, so named because the sequence of an SH2 domain is similar to that of a domain in SRC SARC, another protein tyrosine kinase. Many signaling proteins contain SH2 domains, all of which bind phosphorylated tyrosine residues in a protein partner. GRB stands for growth factor receptor bound protein 2, is an adapter protein with no intrinsic enzymatic activity. Its function is to bring together two proteins, in this case IRS1 and the protein SOS, that must interact to enable signal transduction. So here we have the IRS1 binding to GRB2, and then SOS binds to GRB2 and then to the RAS protein, causing GDP release and GTP binding to RAS. Now, RAS is similar to the G protein that we discussed previously that functions with the beta adrenergic system, and it can exist in either its GTP bound or GDP bound conformation. When GTP binds it, RAS can activate a protein kinase RAF1. And that would be the first of 3 protein kinases in this cascade. And they are abbreviated RAF1, MEK, and ERK. And these form a cascade in which each kinase activates the next through phosphorylation steps. When ERK is activated, it mediates some of the biological effects of insulin by entering the nucleus and phosphorylating transcription factors that modulate the transcription of insulin-regulated genes, and some of which encode proteins essential for cell divisions. The final receptor to talk about are the gated ion channels. These regulate the transport of ions across cell membranes and respond to changes in the membrane potential and ligand binding to specific receptor sites. And the excitability of sensory cells, neurons, and myocytes depends upon ion channels. And these ion channels play very important roles in the nervous system. Membranes are electrically polarized. That means that one side is negatively charged and one side is positively charged. In the case of cells, the inside of the cell is typically negatively charged compared with the outside. And the membrane potential is largely due to the sodium-potassium pump, which pumps out 3 sodium plus ions at the same time that 2 potassium plus ions flow in. This picture shows that the sodium-potassium pump produces a transmembrane electrical potential of about 60 millivolts, where the inside of the cell is negative, represented by all these minus signs, and the outside of the cell is positive. The blue arrows shown here show the direction in which ions tend to move spontaneously across the plasma membrane in an animal cell, driven by the combination of chemical and electrical gradients. The chemical gradient drives Na plus and Ca2 plus inward and K plus outward. Now the K plus in this case is going against its electrical gradient because it's going towards the positive side of the cell. But Cl minus is driven down its electrical gradient towards the positive side. Voltage-gated and ligand-gated ion channels are important in nerve signaling. Nerve signals within nerves propagate as electrical impulses, and this propagation involves the opening of voltage-gated sodium channels. The opening of voltage-gated calcium channels at the end of the axon triggers the release of a neurotransmitter called acetylcholine, and that opens the ligand-gated ion channel on the receiving cell. So in this figure, this is the axon of a presynaptic neuron. And this shows the role of voltage-gated and ligand-gated ion channels in neural transmission. So initially, the plasma membrane of the presynaptic neuron is polarized, where here the inside is negative and the outside is positive. And this is maintained through the sodium-potassium pump, which pumps out 3 sodiums for every 2 potassiums pumped in. There is a stimulus to this neuron, which is not shown in the figure, and it causes an action potential to move along the axon, which is shown with the blue arrow. This causes a voltage-gated sodium channel to open, resulting entry of sodium ions, and the resulting local depolarization causes the adjacent sodium channel to open, and so on. Now, a split second after this happens, the voltage-gated potassium channels open, shown here, allowing K plus ions to exit, which brings about repolarization of the membrane to make it ready for the next action potential. When the wave of depolarization reaches the axon tip, voltage-gated calcium channels open, allowing the entry of calcium ions. Due to the resulting increase in internal calcium ion concentration, this triggers an exocytotic release of the neurotransmitter acetylcholine. So this is, at this point, it's going to leave the cell. Acetylcholine then binds to a receptor on the postsynaptic neuron, or myocyte, causing its ligand-gated ion channel to open. Then sodium and calcium ions enter through these channels on the postsynaptic neuron, and that depolarizes the postsynaptic cell. And then the electrical signal has thus passed to the cell body of the postsynaptic neuron and will move along its axon to a third neuron. This ends Chapter 12. So in summary, we learned about cell signaling, which can be triggered by receptors that sense signals. And these receptors are specific to ligands. They can amplify the signal, they can be modular, and they can become desensitized. We looked at GPCRs, G-protein coupled receptors, which activate interacting proteins. We also looked at receptor tyrosine kinases and receptor guanylyl cyclases. And then finally, we talked a little bit about ion channels and how they propagate nerve impulses. Thank you for watching.